יותר חזק.
מספיק אני...
מספיק אתה צריך לסבול פה. אוקיי. דע לך שזה חשוב, גם לכם התיאורטיקנים.
נכון, צריך לקרוא.
אתם צריכים hands on, זה חשוב. אחרת אתם יכולים... Otherwise you think the cortex is a series of equations.
זה מה שנכון.
Okay. I have to fix the problem from the start. From the basic. Okay. So we have too many students to fulfill all the lectures. So what we're going to do, so only the students who are part of the computational neuroscience program, those are the ones who will present the lectures. Okay? So I just wrote by chance here, I don't know. So Gadi, Roy, and Anat. Anat Shapir is here. Do you have Anat Shapir? So maybe she's not from the computation. She's not. Okay, so we will move her to the non-computational, which is down here. Okay, Guy, who is Guy? גל, אטלן. אחלה. אתה עובד עם עידן במקרה? לא, okay. So Gadi, Roy, and Gal, you will present first on March 9th. That's 3 weeks from now or something like that. 3 weeks from now, the parts of Idan, I already have the papers. I'll send it to you and you can prepare. Next, Chagai, Agmon, Nimrod Levin, and Nofar. Next, Ale. Ma? Nofar is also not? נופר גם לא? לא בקורס אפילו, not even in the course. Anyone here in the yellow which is in the ICNC? שר, נפתלי, צחי, דן, אושרת, לנה, ענת. Dan is not ICNC for sure. Okay, so we're in good shape. אלכסנדר? מהחישוביות? Okay. Lara Lara is here? No? Wow. Okay. Some mistake here. So the Chishuviyut is underrepresented. Seriously. Okay. Shai מהחישוביות, כן. לילך אדלשטיין? גם? Okay, so let's put you up here. The visual system. בוא נראה. מיכאל דורון? כן, נכון? אוקיי. ליאור? אוקיי. רותם מעוז? You will be, לילי. Don't worry. אמיר? גם? רגע, דן ולסקי? אחלה. Who is from the computational and not on the course? Your name? Nora. Okay, and you are in the computer also, and Lily. Okay, who else? נאן? אתה? איפה אתה? למה אתה לא מופיע לי? ואתה במודל? Okay. ניר. Okay, I'll check that. Okay, so basically we have, and all of these people are not, right, from the computational. So the decision is as follows. The computational people will do the lectures, and we have everyone who is not on the ICNC will do a small paper, news and views, on a paper that I will give him. So he will give his comment or opinion instead of giving a talk. I have 2 slots empty, so if you insist to give a talk instead of a small 2-page paper, you are welcome to give a talk instead. Otherwise, what we'll do is we'll do another paper for discussion. The paper that I gave you for discussion for Sunday is going to be in the syllabus of the exam as well. So you should know the stuff well, as if it was a lecture, and it's part of the curriculum of the exam, and we're just going to discuss it in a friendly manner on Sunday, so we're all on the same page. But the papers for discussion, which is this one, and maybe we'll have another one, are also as like a lecture, okay? So, okay.
How long is the talk have to get?
30 minutes, including questions. Okay, so maybe 20 minutes or 15 minutes, something like that. What we'll do today is we'll finish the introduction, and then if we have time, I hope we do, I'll give you what I expect from you from a lecture. So I'll give you a demo of how you should present and put some dgeshim of what is important and what is not, etc., from what to expect. And that's it. And if I won't be able to do it today, we'll do it on Sunday, and then we'll continue the discussion on the Hubel and Wiesel paper. And then we jump into the water and we start. Idan, and then the visual system, somatosensory system, etc. Okay? So I think we're all on the same page and everything is good. Shoot.
Can you elaborate on what you mean when you say discussion?
We're going to put all the chairs in a big circle, and I'm going to put up the slides of the lecture, and we're going to discuss, Gadi, what do you see on figure 1? What did he do in figure 3? What does it mean in figure 4? So it's just a friendly discussion. Where everybody, or several of not everybody, but we should, I will lead the discussion, direct the questions, and if everybody has a different opinion, we will hear it and discuss it, and that's it. Just so we know that we understand the paper, everything's clear, and there's no open questions, and that's it. Okay? This is what I mean by reading the paper. So you should not, be ready for an exam on the paper, but at least if there's a figure on the wall and you never saw it before, that's a bad sign. Okay? You should read the paper. And last year there were students who said, I had neural networks 2 all week. Thank you very much. I didn't read the paper. Please don't ask me anything. And I'm not part of the discussion, which is fine. I just take out 2 points from the final grade. Or 1 point or whatever. Maybe 1 point. I don't remember. But you must attend the discussions, which may be 1 or 2, and then all the lectures. All the rest, you don't have to. And if you're not here for a good reason, let me know, and I'll just make it kosher, and no problem. Okay, so we are still in the very, very basics, just to have everyone in the same line. And of course, if you don't want to come to the lectures of the PIs or me, it's fine, no problem. Huh? But just on the discussions, you must be here. And we're going to make notes, so make sure you sign your name. But all the rest, you're free to, again.
The discussion is the week that the
Papers, yes. Where we have the papers. Now the Moodle is open as well. Sorry, it was my fault. Apparently I can make it happen, and I didn't. So the Moodle is now up. And I put the schedule of the course there. So Physiology I, I think this is your course. So now we have the paper for discussion is up here, this lecture, the full lecture, and the general schedule of the course, which you saw last time with all the talks. General, recent, schedule. I think this is it. Yeah. Okay? So that's the schedule, and you have it up there. Every time there's papers, you must attend. Papers Idan Segev, papers Udi Zohari, papers London, paper Mizrahi, you must attend. So 1, 2, 3, 4. I missed it. Hubel and Wiesel זה 1, 2, 3, 4, 5, 6, 7. 7 meetings you must attend. 7 lectures. All the rest, you're free to go. Okay, where were we? Okay, so we're in the ABC of sensory physiology, and we talked last time about that the first attempts to understand sensory physiology resulted in some psychophysics. We talked about that we can try to quantify perceptions. In humans, it's a little bit easier. In animals, this is also done recently using probabilistic statistics like we saw last time. And now today I want to talk a little bit about the relationship between the stimulus and how it is processed by the brain, which is called sensory physiology. And this, in fact, came about only after the psychophysics because we did not have the tools to study sensory physiology. And only with the inventions of the electrodes, we started to study and to look at the brain by recording it and looking at in more detail. So, first of all, that sensory physiology, when you look at sensory systems, is the most dominant research strategy. On average, if you look around in neuroscience, people follow the sensory information from different stages of processing. We start at the periphery, and we make our way up, and then eventually you find yourself doing your PhD in your favorite location in the brain, maybe 2 locations, 3 locations, I don't know, but we follow the sensory information. The first question is now that if you think you understand the neural circuit, what kind of information would you like to know? Maybe the theoreticians here are good people to ask this question, because when I say, I understand the neural circuit, or let's say you start your project, what is it that you want to know about the circuit that you study? What are the things that are interesting when you study the brain? What do you mean the network? How many cells I have in my network? The correlation between the neurons. Okay, so we're starting to get there. You already know the buzzwords. That's good. So we want to know how many neurons I have in my circuit. Now I have a circuit. You want to study a circuit. How many neurons do I have? That's good. What is the connectivity between the neurons? Gadi, you're a theoretician, right? Or Burak? One of many. Want to be, I see, okay. So now, theoreticians can tell us everything about the network. They know how it really works, but they need also to punch the numbers in their software, right? Or in the algorithms. So the number of cells is good. The connectivity, who is connecting to who. That's also important. The correlation structure, I call it the when-who combination. Who is working when? Because there is the structure of the network, but also, of course, the temporal sequence of events. So you want to know at times, this is the time scale, what happened here, and what this guy did, and what this guy did, and when did they did it, of course. And perhaps one of the most important things you want to know from a network is the input and output function. So what was the input to these cells, and what is the output that came out, okay, from these cells? Now, if you're a theoretician, that's easy because you can imagine what is the input and output. But in real life, so this is something that you want to know. You want to know the inputs and the outputs. You want to perhaps understand something, and you'll see this with Idan, something about the sensory physiology of these cells, because these can impose serious limitations of what the cell can and cannot do in the circuit. Okay, you want to know the synaptic connectivity. This is a very big trend these days, which is revived, to know the exact synaptic connectivity, who is connecting to who. there are different cell types, so it's very important to know if this cell is inhibitory or excitatory or is doing this thing or that thing or neuromodulatory, okay? And like I said, this when-who combination. You want to understand which cell is doing what at what time, and this may be, or part of it is calculating correlation structure in the network, etc. Yes?
Is there some kind of consensus regarding which is more dominant, the cellular level or the connectivity level?
No. So if you'll ask different people, they will think that the real answer is in their hands. And usually we have a very strong microscope on our own study, and we think, that understanding the biophysics of the cell is the most important thing in order to understand. We'll ask theoreticians. They say, oh, no, we need a theory in order to understand this. I think, the truth is somewhere in between, that we want to know, in order to really understand the network, there are many things that we need to know about the networks. The problem is that we still live in the dark with many, most of the 99% of the networks in the brain, we are completely in the dark. Okay? And this is where there's serious limitations of experimentalists. Okay.
Can you please define input and output? What does it mean?
So normally we will go through it, but the input is, when we talk about sensory systems, is the stimulus, the sensory stimulus. Okay? It can be the synaptic input to the cells, which will result in synaptic input to the cells, and the output is normally the spiking patterns of which neuron. Now, of course, I give you only a very superficial list here, because now it's very important if this cell, maybe they have the exact same output, this cell and this cell, but they may go to completely different targets. Okay? So maybe they are not redundant. Okay? If you look at two neurons and they have exactly the same output in time, exactly the same spiking pattern, etc., you would say, this is redundant information. But of course, what is important when you look at neural circuits is who is reading this information. Because, the theoretician can tell me that all the information in the network is relevant, but not necessarily so. Maybe because the real test is the decoder of the code in the network. The decoder here reads what this network has to say, and maybe not all the cells are sending their outputs to this structure. So, this is starting to become very fast, very complicated. And this is why the brain is so complicated, because of this parallel computations and so much possibilities of connectivity and information flow. And we are normally very, very limited in what we see. Okay, we live in single cell and its dendrites and how it computes information, which is fine. Or in a very local network of 10 or 20 cells, etc. But to really understand the system like we understand other biological machines, like the heart or the liver or whatever, the challenges in a neural network are much, much bigger and vast. And I want to give this analogy. I like it because of the big success in biology in developmental neuroscience, or molecular genetics, if you will. Okay? So this is a huge success, and people, it's very easy for me. I'm recorded here, so I have to be careful with what I say, yeah? But it's very easy to be a developmental biologist. Okay? And I excuse the camera here, huh? Put my... And it's not that it's easy on the day-to-day life, but there is a very clear way of doing experiments, and very successful way of doing experiments, very convincing experiments that people are doing. And neuroscience is still like we're still in the stormy adolescence of our lives. Okay, we're still a little bit wild, and everyone's doing. There's no real framework of how to do experiments. And I think with the recent tools in the last 10 years or so, And we're still living a revolution. We are in a place that we can start to do some of the experiments at the level of that the developmental people had in mind. So basically, if we look at cell biology or genetics, so there is a very clear building block. The building block is the gene, is a single gene. And in a very in biology, for example, the human genome has around 20,000 genes, and that's the book. We can start reading from it and start to understand. But that's the basic building block is the gene. In neural circuits, I would say the analogy is a neuron, which is the basic building block of the nervous system. Okay, perhaps maybe a group of neurons, a small group of neurons. Now, if we look at the brain, we have, of course, billions of cells, many thousands of different cell types, etc. So when you want to understand how a neural circuit works, it's now challenging. Like I said, you need to know the cell types, and maybe some are the same cell or not. Maybe we have copies of the exact same cell, so maybe it's groups of neurons that are the basic building block of the computation. And also, genetic networks, they also process information, although on a different time scales, but they also process information. And they process information in the form of protein-protein interactions. Okay? They talk about, I don't know if all of you did some basic course in biology, those are the computational people, but transcription, translation, degradation, post-translational modifications, things like that is the language of the proteins that they work with. Okay? How much a gene is encoded, translated, degraded, can it work or not? In neurons, we talk about spike trains, excitation, inhibition, processes of these neuronal activity, things like adaptations, which you will see during the course, neuromodulations, etc. And perhaps the strongest part of the genetics and cell biology is experiments which are called gain of function and loss of function. So what people normally do when they have some hypothesis about the cell, not only to record which neuron is doing what, this we can do in genetics or cell biology, I don't know, with antibodies or Western blot or whatever, okay? We can say what is a protein upregulated, downregulated, etc. This can also be done in neural circuits. Does a cell, is now strongly activated, less strongly activated, etc. But the real challenge, and that's why this is the strength of experimental neuroscience, is that we can actually do the experiment to test the hypothesis that we have from reading information in the circuit. So in genetics, you just do a knockout, or you take out this protein out, or you overexpress it in, and of course things start to get complicated also very fast because a gene is not an isolated entity, it lives in a genetic network. And I think in the last, 10 years, we're also going into more powerful or more convincing experimental designs by combining molecular genetics, new tools like optogenetics. And this used to be with the lesions. Pharmacology is also used quite a bit these days. So you will, although we will start in the 1960s with Hubel and Wiesel with the electrode, and which we will see the electrode throughout. And even today in our laboratories downstairs, we use electrodes to record from neurons. But the flavor of the experiments is routinely changing into a more powerful designs that can lead us to causal relationships. Okay. Like I said, the classical way is to go with the information flow. We start, sensor information is first gated in the CNS by receptor cells. And therefore, receptor cells provide the first neural representation of the external world. This is the first interface of the sensory world with the nervous system. And this is understood in good detail. Okay? Fair detail, and we will talk about in the different systems what we know, what we don't know. But generally speaking, we can feel good about ourselves. In neuroscience, we have a fair understanding of the behavior of receptor surfaces, many of them, in sensory systems. But again, it gets very complicated very fast. So this information, the receptor neuron information, then flows in serial and parallel streams, and along the way where information now is transformed. And these sensory pathways are recursive, and, they're feedback and feed-forward phenomena, and this is where we fall on our face very fast, and we don't understand. And if you look at sensory systems, so for sure the visual system is the most well understood system, and I would say the chemical senses are the least understood. It depends on in what context, but you'll see as we go along, at least in the olfactory system and the auditory system, which I will talk about, there's very, very big gap between how we understand the receptor cells and how we understand the next stations in the circuit. An important thing to realize is that the nervous system is not a syncytium. I mean, other cells have billions of cells. Okay, the liver has billions of cells. The muscles are composed of millions of cells. But still, it's a different strategy. Again, I don't want to insult people in the cardiovascular system or the liver, studying the liver. But there, life is simple. There are all these cells, let's say, in the liver cells, and they can make contact with, only their next door neighbors. So if it's a 3D structure, connectivity is limited. They can only make contact with their friends above and below, and normally they're also very similar. So if you study the liver, which I just taught in the physiology course, if you look at epithelial cells here, they all share the same design, same principles. They have, a different membrane on one side of the kidney and the other side, and that's it. They're very simple. Same with muscle. You look at a muscle cell, you look at a muscle fiber, you understand one muscle fiber, you can understand the collective behavior of all muscle fibers. They have different properties when they work together, but still, the level of complexity is very basic because the cells are a syncytium. They are very much the same. Here in the brain, it's a much, much, much more difficult task because within the circuit, each neuron can make connections with many, many other cells. It can be hundreds, thousands of cells and get inputs from thousands of cells. Okay? So the challenge, again, stems from the fact that each cell is different or can be different to some extent, and that the connectivity is vast. Okay, so let's start with the receptors. Envision the sensory epithelium is the retina, which you will get to know. In audition, it's the basilar membrane of the organ of Corti. I don't know if you learned about these. If you did, great. If you didn't, stay around. In touch, it's receptors on the skin. In smell, it's the epithelium in the nose. And in taste, we have receptors on the tongue. And each one of them behaves completely differently. And hopefully by the end of the course, you will be an expert on the behavior of the receptors in the different sensory systems. Okay. So, and these come in different flavors. For example, in the cochlea, for example, it's a one-dimensional array of receptors. Okay, and this has to be transformed into, you remember, the latent variables. When it starts with a one-dimensional array of cells in the cochlea, which we will talk about, and it ends in a beautiful music piece. Okay, or something that is recognized as an object. Your mother, your friend, calling to you, I don't know, a dog is barking, whatever. And this transition is exactly this transformation from sensation to perception, but sensation is done by a very basic array of cells in the cochlea. In this case, it's one dimension. In vision, it's a two-dimensional array of cells. In the skin, it's a two-dimensional array of cells. And each one is responding, like we said last time, to a different parameter, physical parameter of the world. Okay? So in the cochlea, it's pressure waves. In the retina, it's light. And in the fingertips or in the skin, it's pressure. Okay. The first thing that we must know, which is shared by all receptors in sensory systems, is that physics in the outside world, whatever it is, pressure waves, chemicals, whatever, are transformed into electricity. That's the first stage in every sensory system. Okay? So whatever variable we're sensing outside is transformed into electricity in the receptor cells, and these are called electrical or receptor potentials. That's one thing. And the other thing that we want to understand is that the energy that we sense outside, which is transformed into electrical potential, is not exactly the same energy, of course. It is transformed, it is amplified, and we are paying big price for it. And the price that we pay is a metabolic price, okay, normally of ATP in biology. That's the price that we pay. And just if you take a simple example, we know that perceptually we can detect even one single photon in the darkness. And the energy calculations for a single photon is 10 to the... This is just a toy example. We are not about to do the mathematics. You can do the mathematics, trust me, much better than I can. But an energy of a photon is 10 to the minus 18 joules, for example. And this one photon, if you record from a receptor cells, okay, will induce a few millivolts of depolarization. And a few millivolts of the depolarization are about 10,000 ions, for example, okay? And the question is, what is the energy of these 10,000 ions versus the one photon ion, which has a 10 to the minus 18 joules. Okay, so one can just do a simple calculation. I don't know if you did physiology. If you did, great. If you didn't, in order to pay for ions moving from one side of the membrane to the other side of the membrane, we use a protein, which is called the NAK pump. It's a protein that sits on the membrane and basically takes 3 ions of sodium, pumps them out, and puts in 2 ions of potassium. And the cost of this work is 1 molecule of ATP, hydrolysis of a single ATP molecule. And just this reaction is the same energy as the one photon. Okay, so it's 10 to the minus 18 joules if you calculate how much is the hydrolysis of 1 ATP. So you can see that we already pay in energy 10,000 times more in order to transfer 10 to the 4 ions. Okay? So basically, it's not the energy that it's not the same energy that it's transformed. The energy hits the receptor cells, and then we start to pay in metabolic language, which in biology it's ATP. Okay? So the basic principle is that the stimulus energy is transformed into electricity. That is the basic language of the brain, electricity. We talk about receptor potentials. We talk about spikes. Okay? And the energy source of the brain is a metabolic energy source. So we pay normally in ATP. This is what we pay. And the process, this transformation from the physical energy into electricity is called stimulus transduction. And this is when the physics of the outside world, as you know them, interfaces with the biology. Okay? It hits the membrane of the cell and is transformed into electricity. So the first conversion of the physical world outside is into electricity, and we pay the money that we use is metabolic. Okay. There are different mechanisms of stimulus transduction. In somatosensation and in hearing, we see direct mechanical coupling. Of the stimulus itself, we will see receptors that are directly mechanically moved, and you see the coupling, this transformation from the energy into electricity mechanically. That's one thing. In other senses, like in taste or in pain, you will see ligand-gated channels. Okay? So there is a molecule that hits a protein on the membrane, and this activation causes some kind of molecular cascades in the cell, or in this case, the ligand-gated, sorry, if it's a channel, the chemical connects to the membrane, and it causes some kind of transformation in the channel, and this causes the ability to transform, to move ions from one side to of the membrane to the next, and cause this electricity, what we call electricity in the cells, which is basically movements of ions from one side of the membrane to the next. In other systems, like in vision and smell, we will see second messenger cascades. Okay, so in this case, the molecule or the photon will hit the photoreceptor or will hit the olfactory receptor on the neuron, and this will cause a cascade of biological events in the cells, which will eventually open ion channels on the membrane and cause the depolarization or hyperpolarization in the cell. Okay, this electricity, the change in electricity. So the mechanisms can be very different for the receptor potentials. Okay, in the book you see, the different energies, the different senses that we will talk about, although we will not talk about the vestibular systems and where exactly do they reside. Okay, but that's very basic. You'll see this in the book. Okay, receptor potentials are then transformed into spiking activity. It can be in the receptor cells itself, in the cell that actually makes the transduction between the physical property and the electricity. You'll see this in somatosensation. Smell receptors themselves, the cells themselves not only transform the molecule into electricity, but also spike themselves. In other places, it will be downstream. Okay, so the receptor cells itself, although it transforms the pressure wave, for example, into a receptor potential, the cell by itself does not spike. The other cells downstream, next synapse, downstream of these cells, only then the spiking begins. We will see this in hearing, we will see this in vision, and all of this is important in order to understand the system that we're talking about. Okay. Neurons, as the basic building blocks of the nervous systems, are basically filters. They are both spatial filters as well as temporal filters. Okay, that's an important point that you will again see again and again throughout the course. Let's look at the visual systems. As you may know or may not know, we have 4 different types of receptors in the visual systems. The visual system in the retina, 3 cones, cone cells, and 1 rod. And each one is basically a filter. So for example, we look at now on the x-axis here on the wavelength in the visible spectrum, which is from the bluish to the reddish, okay, from about 400 to the 700 or so, a little bit less than the 700. And if you look at the behavior of this cell, of this receptor cell, it will absorb light in this spectrum. So the most efficient absorbance is here, let's say 425 or so, and less so as we go along. This phenomena is called tuning width of the cell, and different types of cells have different tuning widths. Some cells, for example, the blue cones peak at the blue, the green cone peaks at the green, and the red cone peaks at the red. Okay? And if you look at rods, which are for day vision, not for color vision, we use them at night mostly. You see that they have a peak at 498. Okay.
The red seems to be at the yellow.
The?
The red cone seems to be at the yellow.
Yeah, that's fine, no worries. It's about 500, the peak is 570 or so. This is called the red cone. It's 564.
So anyway, it's a bit surprising that they're not equally spaced.
Yeah, they are not equally spaced, and you will talk about this with Shaul on how we decode color vision from these three receptors. Okay? But don't worry about the fact that it's called red and it's around the... This is the most red. We have the spectrum goes from blue to red, and the most bluish is the 437, and the most reddish is the 564. And we have one in the middle. Okay, and you will talk when you talk about the visual system how we decode color. I hope we'll talk about color vision from these receptors. Now, of course, you can see there's a problem here, because if you would try to extract the information only from the red cone, let's say this is the number of spikes. This is not. This is the absorption, but let's say it's the number of spikes or potential that this cell responds to. You see that at the same level, also at 500 and at 620, this cell has the same number of spikes. So it will not be possible to know if we are reading information only from this cell, we're going to have a hard time to know if it's 500 or 620 because of the receptor field of the cell. And this is why we need the combinatorial information from other receptor cells, of course. Okay? And this now is starting to become, our neural network. Let's try to understand. Okay, now the input was, I don't know, green light. How is it encoded in the cells of the nervous system? So this is at the heart of sensory physiology. What is the input? It was green light in this case. Which neuron responded to the green light and in what sense, and what is the receptor field, etc., etc. Okay? So this is the type of things you will start to look at when we look at sensory systems. Yes? No, on the contrary.
So, two questions. What's the scale of the vertical axis?
This is maximum absorption, which would be 100 or 1 at the top. Okay?
And are they normalized relative?
In this case, they are normalized. The absorption, yes.
Relative to each other?
No, not relative. No, they are relative to their own...
The maximum of the red cone seems a bit higher.
Yeah. So it's a little... Each one is to its own. I don't know if... And it's not very important at this stage of the course.
And the curve of the rod, why should it have a different rate, different response to...
What do you mean? That's the property of the rod. This is the cell, and when you hit it with 498 or 500, there is maximum absorption here.
But you said in common knowledge that rods are like more for night vision.
Yeah, but you are running too fast. Okay. When we get to the visual system, we'll talk about that. We're just now very basic principles. Okay. What I wanted actually to tell you is that many cells have receptor fields, and you can talk about things like tuning width and best frequency of the cells, etc. Okay? And this is an example, a real-life example, from a neuron that we recorded in the auditory cortex, for example. And on the y-axis, you see the stimulus, which is in this case frequency, from 3 kilohertz to 80 kilohertz. And on the y-axis is time, from 0 or minus 50 milliseconds to 200 milliseconds or so. It doesn't really matter at the moment. And you see that this cell does not respond at all to 3 kilohertz. He starts to respond here somewhere, and he has some kind of width to its response. So he will respond to a given width of cells. Perhaps its best frequency is here, or here its maximum frequency. This design is called the raster plot. You should know, right? What is a raster plot? Okay, so this is called the raster plot, where we show the spikes of the cells, and each repetition here is one trial. Okay, so here we have, 20 trials for each stimulus, And you see we have the spontaneous firing rate is rather low. You can see it here. And this cell responds to this region of the stimulus space. Another type of representation that you will see throughout the papers you will look at is something called post-stimulus time histogram, a PSTH, which is basically a collapse in time of the raster plot. Okay, so basically this is a collapse in time. You have some noise or some basic spontaneous firing rate, and this cell, the stimulus onset was here, responded, and died off a few hundred milliseconds later, or a few tens of milliseconds later. Yes?
Can you go back to that previous?
Yeah.
So, they're not symmetric, they have a longer tail?
Some receptor fields are symmetric, some receptor fields are not symmetric. Okay, and there are some reasons for that. Okay. Let's look at the real receptor field of the cell. For example, in the auditory system, we talk about frequency response areas, and then we'll take a break. It's the number of spikes. Okay? So it will be the spike rate, for example, or the frequency of response. Okay, number of spikes per bin, and you can bin it differently. So basically, in the auditory system, we, for example, have these receptor fields. So you will look at a receptor field which looks like this, which is now a little bit different design. So you take the frequency. In the raster plot, you saw the frequency, and now we collapse it on this axis from 3 to 80 kilohertz. These are different frequencies. And this is now the intensity of the response of the neuron. How much it, the intensity, sorry, is color-coded. In this case, it's from a maximum, the maximum firing rate of the neuron. In this case, it's normalized to itself. And this is the different intensities of the stimulus. So this is 72. It's a strong stimulus, less strong, and less strong stimulus. You see this neuron does not respond at all to any of the frequencies in the low intensity, but as you increase the intensity, you start to have a shape of the receptor field. And again, this does not have to be symmetrical. In many cases it is symmetrical. In many cases, it is not symmetrical. Okay, normally there's a sharp cutoff at higher frequencies. This is only for one neuron. And now let's go through the way we calculate it. Okay, so basically, we started. This is the raw data, the spikes, and this is the PSTH on the top, the PSTH, the spikes, and we can construct from all these frequencies. Okay, this is just one. The PSTH is a very redundant way of showing things because you lose a lot of information. Here we see we have a lot of response in the low frequencies. You can see it here as well in the low frequencies, and this dies off very fast, and you get very low responses at high frequencies. So this is how we calculate this. Then we run another series of experiments on the same cell. And this is the PSTH, the raster, and the response area. And we do it for the lower intensity and lower intensity. Okay, this is real data. And this would be a receptor field, or in audition, we call it the frequency response area of the cell. This is the types of structures that you will have to look at. Okay, the receptor field of the cell. Now, because different neurons now have completely different receptor fields or tuning curves, this combinations of low threshold, high threshold receptor fields, narrow receptor fields, wide receptor fields, these things, these properties of the receptor fields increases the dynamic range of the stimulus itself. Okay? Now the transformation, I hit this thing with a green light, but then, you know, this cells fire a lot, this cells fire a little bit, this is not firing at all. This will now respond to everything. You know, now maybe your stimulus is not 536, but from some kind of band pass stimulus from 480, I don't know, to 530, and now, you know, this cell will respond such a way, and this will respond in such a way, etc. And the role of the decoder is to understand what the stimulus was. I'll leave you, we'll go to the break with this one example, again, a real-life example, just to show you or to give you a teaser that when you go into the brain, in higher stations in the brain, things start to become much, much, much more complicated. So these are, for example, 15 different neurons responding to exactly the same stimulus. It's a very long stimulus, maybe 10-second stimulus. In this case, it's vocalizations, recordings from the auditory cortex. In this case, in my lab, you see the first cell on the top responds almost to nothing. This is 20 repetitions of the same stimulus, okay, for each cell. This cell, for example, responded to the onset of the call, some kind of noise here in the call. This cell, the first one, number 15, responded only to the offset of this call. This neuron responded to each and every syllable in the call. Okay? This neuron responded very strongly to syllable number 7, very weakly to syllabus number 3, and here also with 20% probability, and here with 100% probability. This cell responded with 5 spikes, and this cell responded with 1 spike. And this same cell responded with 1 spike to syllable number 3, and 7 spikes to syllable number 18, etc., etc. So you see that the same cell, and the things that govern the properties of these cells are the biophysics of the cell, okay? All kinds of things like adaptation, time constants, synaptic depression, etc., etc. When we will look at receptor cells, things will be a little bit more intuitive. We understand them very well. But when we go to cortex, and this is how a population of cells in cortex looks like, now you must tell me, as a decoder, what was the stimulus here? Did you hear this sound, or did you hear another sound, etc.? And that's the big challenge. We're very far from really understanding how we can extract the perception out of these types of codes. Okay, so let's take a 15-minute break. Go back to history. זה עכבר, כן.
רק כל שורה שם זה תא אחד?
תא אחד ו-20 חזרות.
כאילו כל...
חזרה הבאה, הוא יראה, ואז מחשבים את הפרובביליטי בוואי עצמי. כמה פעמים מתוך העצמי הוא יראה. זה סילבל, זה העברות בתוך עצמי. זה זמן, זה 200 מילישניות, וזה רסטר.
אוקיי, זה גם עבור נוירון.
עכשיו אני רוצה לבנות רספטופיל של הטיפ.
לא, זה ברור לי. אז תתחיל עם הזמן, מה שקרות זה...
כל אחד כזה זה טרייל אחד. רצתי ונתתי גירוי פה בזמן אפס של 80 קילו. הוא לא ירה בכלל. עשיתי זה 20 פעמים. אחר כך נתתי ב10 קילו, והוא ירה ב5 ספק. זה פשוט סטאק של המון חזרות. הגירוי פה הוא הגיב 7 ספייקים.
אני מבין, אבל אני מנסה להבין מה בדיוק המשמעות של נקודה אחת שאתה עושה פה נקודה ככל ספייק. אבל לא יכול לעשות איזשהו ספייק כזה, לעשות ספייק בצורה כזאת וספייק. אבל לא יודע, אני ראיתי שיש כאילו כל מיני סוגים של...
לא. אתה יכול לראות 5 ספייקים באיזשהו טמפורל סיקוונס.
יש איזשהו, אוקיי, זו תגובה בינארית כן או לא, אבל יש איזשהו גיוון ב...
הגיוון הוא בטמפורל סיקוונס. אתה יכול לראות ככה, או שיכול להיות רצוף, או יש לו כל מיני אופציות. אבל השפה היא שפה של ספייקים.
מבחינתך הם כולם בדיוק
זהים. כי זה ספייקים.
כן, אני יודע.
תדבר על ממרי פוטנציאל, זה משהו אחר, זה כבר סיגנל אנלוג. זה ממש סיגנל דיגיטלי. זה ספייק.
עכשיו נניח כמה שניות יש לך פה?
פה יש לי 200 מילי שניות.
200 מילי שניות.
הגירוי הוא 50 מילי שניות, מ-0 עד 50.
אוקיי. אז בעצם כל שורה פה מייצגת טרייל אחד. ואז בעצם זה חוזר כל הזמן, נכון?
נכון, אבל זה חוזר, לא. יש לי 20 חזרות על 80, 20 חזרות על 34, 20 חזרות.
אבל התדר, מה זה הסטימולוס שלך?
במקרה הזה זה פיוטון. בדיסק זה פיוטון של פרקי.
של תדר.
כן.
ואתה בעצם משנה את התדר.
כל גירוי אני משנה את זה.
אני משנה את זה, ואז אתה מסתכל מתי מופיע ספייק.
בדיוק, אבל אני לוקח את כל הספקטרום ואני אומר מ3 עד 80 קילו, זה תחום השמיעה שלו. בוא נניח? כן. תחום הראייה, תעשו אותו דבר. פולס של אור, מ-400 עד 800, ניתן פלאש אחד. כן, נניח מ-400 עד 800, ניתן פלאש, תראה שלא היה ספייק. פתאום אתה מתחיל לראות ככה. ואז אני בונה רספטר פילד לספק.
אתה יודע בוודאות פה שאתה מגיב מה-20 בערך מ-15 עד 30.
נכון, אבל הפרובביליטי שלו להגיב הוא הרבה יותר גבוה בנמוכים.
כן.
אוקיי? וזה הולך ודרך. אז יש לו איזשהו מה שנקרא bandwidth.
bandwidth של הגב. בונה את הפאוור.
ולכל ניורון בדרך כלל יש רספטר פילד ויש לו איזשהו...
מה ההגדרה של רספטר פילד? זאת אומרת, התחום התדרים שהוא מקיף. בכל חוש זה יש עוד. נכון. בראייה, רספטר פילד זה איפה בעולם הוא הגיב. במיקום המרחבי בעולם. ואנחנו רואים בוויזרד גיבל בוויזרד. הוא הגיב מאזמוס כזה או כזה, או מאזמוס כזה או כזה. שמיעה מדברים על frequency response area, באיזה תחום תדרים. בריח מדברים על molecular receptive range, איזה מולקולות יהיו. כל אחד עם אז פה יש לנו number of spikes, נכון?
כן.
וזה ציר הזמן שאותו דבר.
זה בדיוק ציר הזמן. PSTH עושה קולאפס.
כן.
במקרה הזה, פה אני מראה לך את כל הדאטה. פה הראיתי 4 דוגמאות שונות, כן? זה הראשון, רק שניסיתי להמחיש, ואז עושים את אותו ניסוי, אבל זה עוצמות נמוכות יותר.
בתדרים...
אותם תדרים.
אז אני לא מבין.
אותם תדרים. חזרתי על כל התדרים, מ-3 עד 80 בעוצמה החזקה. וזה מה שקיבלתי, רסטר ו-PST. עכשיו אני...
עוצמה שמה? של הגירוי?
כן.
עכשיו אני... יש לך בעצם שתי דברים שאתה משנה. זה התדר והעוצמה של הגירוי.
כן, עוצמה ותדר.
כן, אוקיי, ועכשיו אתה יכול לזהות איפה יש לך פאוור.
עכשיו בחוש הריח בראייה אתה גם תראה, משנים תדר ומיקום בעולם המרחבי. אני לא יודע, גם פה יכול לשנות פיץ' ותדר. אפר ותדר, לא מה שאתה AM ותדר, מה שאתה
אתה יכול... אתה יכול להגיע למצב שיהיה לך B-model distribution, שיהיה לך בעצם... שתי פיקים, בדיוק, יכול להיות? ממה זה יכול לנבוע?
מה זה נובע? זה נובע אף ורק מבחינת הקונקטיביטי של התהליך. ברספטורס לא. זה רספטורס.
לא ברספטורס, זה
בניורונים. בניורונים מהזה אתה רואה, פיקים משתי דבשות, יש כל מיני דברים מוזרים.
כן, אז מה זה אומר? מה זה אומר שיש לי דימות?
זה אומר שיש לך איניביציה במקום מסוים. נניח זה התדר. ואתה עכשיו מגיב, הוא מגיב מאוד חזק פה, והגיב מאוד חזק. זה אומר שפה יש איזושהי איניביציה.
ברגע שאתה נותן לו, אני לא יודע, 10 קילו הרץ, אז אתה מפעיל את האיניביטורים שמעכבים את הנניח.
ואם זה פשוט יורד בצורה ארוכה, זה אומר משהו, זה עדיין, זה לא בלי מוד, זה לא אומר כלום?
זה מה שהם אמרו, לפעמים התאים יהיו לפעמים הם יהיו ככה,
תלוי בפריש ובריאש. ומה אינטרספייק?
כן, לא, אני מרגישה כאילו כן צריך...
יש לך ספייקים נכון? אתה מחשב
לא, היינו הולכים להגעת... אומנם אני יודעת שאין לי קשר... לא.
אתה יכול לעשות את החוקרים, אתה תראה את הוויז. זה מה שזה ייתן לך טימור.
באמת לא דיברתי איתם, הילדים שלנו.
אתה מערבב
אם יש לך, אתה יכול לדבר כזה, או דבר כזה.
מה הקשר?
כולם מסוים ולא הייתי באף אחד לשפר? לא, אני אבוא גם לשיבה, אני אומרת, זה לא שאני בתשובה שחורה לדעת, שלא הייתי באה לשיעור, אבל...
וזה באחד, אז לזה שהקונסידריישן שלו יותר נמוך. זאת אומרת ש... כן? זה מה ש...
חודש שלא יש לך רגע. אוי,
איך זה בעצם קשור
בדרך כלל כל תא יש לו איזשהו דווקא באינטרסטים אינטרוול יש להם תכונות מאוד מסוימות, לא יכולים להיות משוגעים לגמרי. בדרך כלל כשעושים ספייק סורטינג מזהים אותם אחת השיטות לפי זה בדרך כלל יש לו דיסטריביושן מאוד מסוים.
אני לא חושבת שזה זה הולך לאיבוד.
להבדיל בין כמה... תודה.
לא יודעת. קיצר, אתה גם נכון? כן, אני שלמה עם זה. כן, אני אמרה שכותב במחשב, נשלח לי וגם רציתי לבקש ממי שיעלה את זה גם, כי אני מה לא רוצה שזה אני יודע ממש לא. אבל אני צריכה לראות את זה, לא? למרות
Okay, so we go on. As you will learn, science is changing quite a bit with implementations of new techniques. This is very clear in the in all biology in general. But basically now you are living a revolution, although you don't feel like it. You are living in the middle of a what? What kind of revolution? No? Okay. Where methods are continuously improving and making new things possible, and changing the way we look at the nervous system. So we're going to see, we're going to start with classical techniques, basically mapping the brain with electrodes, the work of Vernon Mountcastle, which started in the somatosensory. The first person who stuck electrodes into the brain was Vernon Mountcastle, revolutionized neuroscience, and Hubel and Wiesel picked up Mountcastle's methods and looked at the visual system, and we will see, we will talk about that a little bit as we go along. These days we can do fancy things. What you see here is a mouse running on a ball in a virtual reality environment, okay? And today we can record from cells or image the cell while the mouse runs, and then, you know, Mickey Mouse comes from the right, and he has to turn to the left, etc. So while the animal performs behavior, we can interact with the animal. And of course, I mean, you saw last time I gave you the example of Yitzhak Fried, of the patient who was recalling The Simpsons. Here we can, in mice, we can do, or different animals, we can do different things, which can take us, can bridge this gap between the sensory physiology and the behavior, and eventually what we want to understand is perception as well. So because the language of the nervous system is electricity, the most powerful techniques are electrical recordings from the brain, and there are many variations of electrical recordings from the brain. Starting with Bert Sackman in the late 60s, we can now record from single channels. These are methods that are called patch clamp techniques, which can record a very fast event of opening and closing of ion channels. One can record from neurons spiking activity. You should know what an action potential is. You can record from neurons in different modes, in voltage clamp mode or current clamp mode. So you can get the, in this case, an excitatory postsynaptic potential, which is called the EPSP. These are the few millivolts which I told you that a photon will hit and excite the cell to get depolarization. And you can also, this is in current clamp mode, and you can also voltage clamp the cells and get the currents from the cells. Okay, so this would be equivalent to this. This would be an excitatory postsynaptic current versus the potential. Okay, this is based on Ohm's law. V equals IR. You can extract the voltage or the currents. Normally, you can stick an electrode into the cell and get or a single spike, or you can get bursts of spiking activity. Cells a lot of time will burst. Going upward on the scale is depolarization. Going downwards is hyperpolarization, of course. Other modes of recordings that you will see are local field potentials, where you basically record from the extracellular environment of the tissue. You stick an electrode into your favorite tissue and get a recording from populations of cells. There are many, many very smart people who are trying to understand what is happening inside the local field potential recording. Not a very easy signal to understand, although there are all kinds of methods that one can extract the meaning of these signals. Also, EEGs, electroencephalograms. You can do a non-invasively record in humans. I think Leon Duell, for example, is doing these kinds of recordings. from people, and this basically sticking a lot of electrodes in different places to understand the signal. Okay, so the advantages of the electrical recordings is that it's the real language of neurons, very good signal to noise ratio, especially in the single cell recordings, not so much in the EEG, and in all cases they have superb temporal resolution. Yeah?
What about non-spiking neurons? How do we usually measure them?
That's a very problematic thing if you want to record extracellularly. If you record intracellularly, then it's not a problem. Then you can go into the cell and just record the differences in membrane potential, for example. So if you have, in the visual system, we will see that the receptor cells are not spiking. You only have current changes. And now, if this happens in a lot of neurons, maybe you can sense something in the extracellular environment. But this is very hard. The intracellular signals are very big. Okay, they come in millivolts normally. So a spike can, go from -60 to 10, 20. So it's a huge signal. Signals from the outside, when we record from the outside, it's much more difficult because the sizes of the currents are very small. Now, if a neuron spikes, he also creates some current outside. If you have some kind of difference in the membrane potential, it's extremely difficult to see it from outside. So you have to go inside the cell. But silent neurons are also something where there are silent synapses, all kinds of interesting things. I don't think we will get to that level in this course. Okay. And now, recently, like I said, we'll go from the old to the new. This is a new trend of methods, which you will also see is using microscopes, fancy microscopes, to record from the cell. What you see here is, again, in my favorite part of the brain, which is at least one of them, in the auditory cortex in this case. And what you see here is a focal plane of a 2-photon microscope of a live mouse under the microscope. And you see a single optical plane. This is the surface of the brain, about -50. And you see in red is staining of astrocytes. And in green, you will see neurons. And what we do is we change the focus of the microscope as we go along. Ready? Buckle up. And we go deep. We go down. You see the astrocytes going down into the brain, down into the brain. This is a live mouse. And suddenly you see the neurons in green. Okay? We go down until it becomes dark again. But basically, what optical physiology lets us now do, the mouse is under the microscope. We can change the focus. Let's say we like this region. We think these neurons are interesting. And now we can study only these neurons. We stop optically in a single optical plane in the brain. And we have, 10, 15, 20 neurons which are now accessible optically by a microscope. And we can now place stimuli to the brain. And you get results like this, so something like this. This is now 16 neurons, which we image using 2-photon microscopy in the intact brain of a live mouse. And you can see now we get the activity patterns of single neurons. Now, what is... Chagai. In this case, so before I talked about spikes, which are binary signals, if you will. Here, it's a little bit more analog signal, and you will see that the different sizes of the signals. Some will be small, some will be large. Now, we think that in this case, this is calcium signal. It's an ion in the cell that we monitor. We think, or we try to claim, that a large calcium signal is equivalent to many spikes, and a small is equivalent to a few spikes. But generally, it gives you now the option to record from many neurons simultaneously. And this is great ammunition for the theoreticians, okay? Now they can plug in the activity of many neurons simultaneously, and like you said, to calculate, I don't remember who mentioned, to look at the correlation, or what I call the who-when combination. So now we can look, now again, it's limited because the neural networks are tens of thousands of cells, but now I can look at, like you saw before, in this example. We can go in, and now we can pick our favorite cells. Actually, I just want to show you also. Oops, sorry. Actually, you see, I don't know if you see it very well, but there are also cells here. I don't know if you can see it. You can see the shadows of the cells. Don't see it very well with the light from there. Anyway, so basically you get all the volume of the tissue of the brain, and you see the shadows of the cell, and now you can stall on a single optical plane, and now you get these 16 cells, etc. Now you can start to calculate in the computational neuroscience group, signal correlation, noise correlations, all your favorite correlations. This is changes in the fluorescence of the cells, yes. Okay, and we may talk about this, but again, because you're going to read papers, either electrophysiology, but we sin a little bit in this course because we give you the very basic, very, very basic, and then we push you to the state-of-the-art projects in the field. This can be done these days with awake behaving animals. Especially in rodents, you can implant all kinds of machines. In this case, it's a portable 2-photon microscope. And again, you can look at neurons at the single neuron level, at the basic building blocks of the brain, while the animal is behaving. Okay. Sometimes neuronal activity is nicely correlated with perceptual performance, like in the somatosensory system. No, but most of the time, so in this case, the amplitude of skin indentation, and this, what is this? I don't remember what's on the y-axis here. Anyway, probably the feeling, the perceptual score. So you see, I can tell you, the person in this case has to tell if what was the level of indentation from X to a maximum, and there's a very clear linear relationship. And in this case, also the neurons, the receptor neurons on the skin, you will get... so this is the perceived intensity, sorry, and this is the number of spikes probably. Sometimes there is nice correlation. But in many cases, there is not. So if you look at, again, the somatosensory system, you can have different types of receptor cells. So in this case, this is different intensity of the stimulus, from 270 micron indentation on the skin to 570, 880, 990, increasing intensity of the stimulus. You can see that this is the spike rate of this receptor cell, and it increases the firing. As you go higher, you get more and more firing, like you saw in the first graph. Okay, these types of receptors are called slowly adapting receptors, and they are very good in decoding the information of the intensity of the stimulus. You see, because the low, especially if you look late in the response, so you can see a linear increase in the firing rate of the cell. But some other new types of receptors, which you will also get to know, are rapidly adapting cells. Okay, and they encode something completely different. They will, if you give a square pulse, they will spike only in the beginning, at the end of the stimulus. But if you now give a ramping pulse, they will spike very strong if the ramp is high, and as you decrease the ramp, they will spike less and less. Okay, and of course, if the stimulus doesn't change anymore, it's rapid, it's still on, you'll get no spiking, while in these cases, you will get spiking, which is relative to the intensity of the spike. So you see there are different, already from the beginning, there are very different strategies of neurons to encode information. And each one will encode different things. Okay. We have all kinds of limitations when we look at receptor cells. First thing I told you is the spatial filtering. We saw it in the visual system, where some neurons are responsive only to given frequencies of light. Also, some neurons are only exposed to a given part of the world, in vision, for example, In auditory, they will respond only to some frequencies. In the skin, of course, you know, the cells will reside only in a given place of the skin, so that's a limitation, a spatial limitation. And in many cases, not so much in the chemical sense, we will talk about that, but in other senses, in most other senses, there's a very clear organization, topographical organization of the receptor cells on the sensory epithelium. And this will determine to a large extent, the responses. Okay? In chemical senses, it's a little different, and we'll talk about that when we will get to there. So this is very clear or intuitive in skin cells, in the somatosensory system. Each cell will have its sensors on a given spatial extent of the skin. And when you look at the stimulus itself, you know, if I push the skin, indent the skin here, so this cell, for example, will be active, and the rest will be silent. So we're going to have like an image of the stimulus, or the scene of the stimulus, by decoding the information from all of the receptors. So silent neurons are as important as active neurons in order to reconstruct the scene of the stimulus. Okay? The information would need to come from a population of cells, and very, very rarely you will see information that is encoded by a single cell. A single cell can instruct or impose information, but to get the full scene or to understand the stimulus, one has to reconstruct information from many, many cells simultaneously. Okay. Now the question of resolution. What is the resolution of the system? It depends on two things. One is the density of the receptors, okay, the number of receptors per unit area, but also the receptive fields. So a neuron can have a very narrow receptive field, or it can have a very wide receptive field. It can, see, so we have, I don't know, in this case, this piece of skin has, I don't know, 2, 4, 6, 7 cells. Each one has a small receptive field, but maybe we can encode differently the information if we had 3 cells with large receptive fields, or if we have 100 cells with very tiny receptive fields. Okay? So these are things that one has to take into consideration when you want to understand the resolution of the system. And you will see different places in the brain with different resolutions. This is again intuitively explained here, just by pixelation of an image. If you have less receptors, in this case they are sensitive only to the region of the image, you can get only so much resolution. The more receptors you have per unit area, the better resolution you have. This is standard, intuitive, and no problem. Some regions of the body have higher density of receptors. Okay, this is the famous caricature where, you know, our lips are very sensitive in the somatosensory system, and also the fingers, the face is extremely sensitive, and other places of the skin is much less sensitive, like the receptors on the back. In high school, this is a very classical example where we show kids to try to resolve 2 different spots on the skin, and you can do get very good performance with fingers, but very bad performance with on the back or the back of the hand, etc. Okay, and this is due to the difference of density of receptors per unit area. This is true also in vision. In different parts of the retina, you will see different sensitivities. So at the fovea of the visual of the retina, you will have a high density of receptors, and this means that our resolution in the center of the field is higher than at the sides. So this is also something you will get encountered throughout the course. See, I won't have time to get to the demo. Let's see how much stuff I still have. Okay. One thing which is common to all sensory systems is that the initial steps of the information or the initial steps of sensory performance is to deconstruct the stimulus into its basic stimulus components. And as you go up the hierarchy, you will see more and more complex features of the cells. So you will never see a Simpson cell in the cochlea, okay? It's a neuron in the cochlea or a cell in the cochlea that will respond to a high-level feature. The brain works in such a way that it first deconstructs the stimulus into its basic properties, wavelength, frequency, etc., and only then, as we go higher in the brain, you will find neurons that respond to more complex stimuli or richer sets of stimuli. Now, this is in contrast. You remember that what we talked about when we saw last talk about the experiments of Ant-Riesman, we can identify perceptually faster, higher cognitive regions. You remember I gave you the exam of the sign that you had to read, and you skipped the is, the letters, and when Ant-Riesman said, to identify the X in these matrices, you can identify higher cognitive things faster, and to go back to the details takes actually more time. But if you look at the hierarchy of information, that is, you stick your electrode into a neuron and ask what is the responsiveness of this neuron, you will see very low levels first, and only then, as you go up high in the hierarchy, different circuits, you'll see more and more complex features. This is a very common general principle of sensory systems. Okay. Spatial sampling. Spatial sampling is limited by rules of physics, and basically what you want to know is that in order to reconstruct a given property or a given resolution, given stimulus, you are limited by many factors. So for example, if we want to reconstruct a sine wave, a 5 Hertz sine wave, we are limited by what is called the Nyquist criteria, which is true for band-limited signals. Again, we don't want to go into this too much. Maybe if you do our advanced course in imaging and microscopy and optics, we go into this a little bit more detail. But the bottom line is that if you have a stimulus, let's say a 5 Hertz sine wave, you cannot get a resolution or reconstruct this 5 Hertz sine wave if you sample it too low. Let's say if you sample it in 1 Hertz, the best you can get is this dotted line. Okay? Clearly, if you want to reconstruct the 5 Hertz signal, you must reconstruct it at a higher frequency than this 1 Hertz, because otherwise you get aliasing of the stimulus, you get distorted stimuli. You would think that it's the dotted line and not the 5 Hertz line. And the Nyquist criteria says that you must sample a given stimulus at twice the rate of the highest rate of change. So if this is a 5 Hertz stimulus, you must sample it at 10 Hertz. And if you sample at 10 Hertz, you can get a perfect reconstruction of the 5 Hertz signal. If you sample it at higher rate, you're wasting energy. Okay, there's no need to sample at higher rate in order to get this type of resolution. And in many cases in sensory systems, you will see that we are bounded by the Nyquist criteria, and we can only sample so much because of the density of the receptors, because of the overlap of the receptor fields, etc. Because these are the sampling of the system itself. Okay, the resolution is bounded by the sampling rate of our system. Now, this is the normally regarded as the spatial sampling. Okay, we need twice as much of the highest rate, or 1/2 of the spatial extent. And I don't know if we have time for this. Not so much. Let's skip that. Anyway, one can calculate what is the, let's say if you look at the retina, This is an image of the retina and the photoreceptors on the retina. And the spacing of the receptors is about every 2 microns or so in the highest region. In the fovea, you'll get every 2 microns, roughly, you'll get a photoreceptor that samples the world. Okay? That's the highest density. And indeed, if you calculate what are the physical limits that one can look at, you'll get to numbers. And if you will go back to the slide, you will get that the minimum spot that we can get on the retina is 1 micrometer of light. Okay, we cannot do better than that because of the physics of the eye. And there are other distortions to the eye, and indeed you can see that the biology is well tuned to the limitations of the physics that are imposed by the physics of the eye in this case. Okay, and this is probably the result of millions and millions years of evolution, that now we have 2 micron spacing between each receptor cell in the retina, and indeed it fits beautifully the eye, the size of the eye, in this case the human eye. If you look at other humans, for example, are as good as it goes in terms of visual resolution. I think you can see, you can differentiate 2 apples cheek to cheek sitting next to another from a kilometer away in good eye vision. And humans, if you look at the animal kingdom, are as good as it gets. We have some competitors in the birds. The birds of prey are extremely good, and they have smaller eyes, but still they have a huge density of receptors in their eyes. And we are very good in vision. Other animals are very good at other things. I mean, we cannot compete with rodents or dogs in smell, for example. Okay, they are much better than us. And as we talked about last time, each animal has its own advantages, and when you go far enough, each sense is species specific. Okay, and now we have limitations of the system, physical limitations of the system. So for example, spatial vision, somatosensation are normally limited by the Nyquist rate, which we talked about before. Okay, whatever you can sample, you can do as the best you can do is twice as much. Okay, so if you look at a signal, I don't know, 5 hertz, you have 5 hertz sampling, you can get your resolution is 10 hertz or so. Okay, that's the physical constraint. This is true for the sensation. You know, how good you can differentiate 2 points in the skin are limited by your sampling density. But some of the you will see that some of the perceptual performance, work with hyperacuity. So we can perceive actually much better than the physics imposed on our senses. So for example, if you look at the bandwidth of receptors, which is about 1/4 of an octave, you would say, okay, I can do only so much with my auditory system. If I put now on the cochlea receptors that are responsive to frequencies at a given frequency, which is whatever, it doesn't matter, X, I don't know, here it's 130 hertz, now this cell responds to 60, this cell responds to 390, etc., etc., okay? So you have a 130 hertz resolution. But perceptually, we can perceive 5 hertz from 1000, okay? This means that we are working in a mode which is called hyperacuity. We are exceeding the best performance that our senses limit on us. Color vision is a good example. We have 3 receptors, but we can see hundreds of color hues. And this is also a way to extract in a smart way from our sensors better acuity of the system. Okay, and we do it by sensory coding, by how neural networks work in the brain. So we can extract, actually in several cases, we are limited by the physics. In other cases, the brain can extract better information than what is imposed from our senses. Yes?
Done by understanding of the environment, or at any environment we will be able to use things that what the receptors allow.
I think it's in. I don't understand exactly what do you mean by assumptions on the.
I mean, it is enabled by heuristics that the brain assumes.
Yeah, I'm not sure I completely understand. It's done by pooling information from many cells together, and when you do that, you can extract actually better resolution than what the receptors can tell you. I don't completely understand. Okay, and these are the spatial limitations of the system, and there are also temporal limitations of the system. Okay, and there are two types of temporal filters. So neurons are spatial filters, and there are also temporal filters. And we are limited from above, and we're limited from below. From above, we're limited by phenomena like time constants, or the rates that are associated with the transduction that we talked about before. Okay, so I can detect a smell or a photon only so fast, because once it hits the photoreceptor, it has to get transduced, and now when a second pulse comes in, there is this time delay that we have to take into consideration. So from above, and of course the time constants of the membranes themselves, the neurons are RC circuits, and they are limited by these cable properties of the neurons. So all these time constants impose a limitation that a cell can fire so much and be accurate only so much. And from below, we have also processes which are inherent into the biophysics of the cells, which limit us. And these are processes like synaptic depression. You can excite a synapse, and then when the next pulse comes in, sometimes the synapse will be depressed, so the next pulse is weaker, and phenomena like adaptation, which you will encounter as we go along. Okay, but there are limitations from below and limitations from above on the response of the cell, on the cells. Generally speaking, temporal filters. So again, there are limits on the temporal resolution. Vision and somatosensations are limited to about 100 hertz, and the auditory is very special, that it can do things like phase locking up to 10 kilohertz, and I think we will talk about that. Okay, and envelope coding, things like other decoding processes of the auditory system have different temporal resolutions. Okay, more or less we're done with the introduction. Next, any questions on the talk? Simulation of what? Yeah. So the simulation was first of all, we have 2 minutes. The simulation is me giving you a talk, a 20-minute talk on a paper. I want to show what I expect from the students, and how to give a talk and what to give in a talk. I will give you an example of a paper that is coming out in 2 weeks, our paper, and I want to show you what is in the paper and what is a good presentation, what I think is a good presentation of the paper. Okay, so this is I don't know, a 9-page paper. This is something that you will work at, and you see it has 7 figures and supplemental figures, and I want to show you what would be a friendly way to show this paper. Okay, so this is the paper I'm going to show you. And eventually I'm going to put this paper on the website and also my talk on the website, so you can see that the main thing I want to tell you is that it's a different skill. It's a new skill. You know, writing a paper and putting all the details in is one skill, but presenting it to people who are not in the field is a completely different skill. And the most important thing is to know how to extract the important information from a paper and present it in a gentle way to your audience, so it will not be too painful. Okay? Now, some Americans are very good at this. They know how to deliver, say the right words. It's very smooth. It's very common. Americans are very good. Israelis need to learn a little bit more. So this is part of this course that we, and actually a very important part of the job is to know how to convey a message. Okay, so we're done with lecture number 1. Next time I'm going to give you a demo on what are the important things I want from